Well, let's get started. Good afternoon and welcome to the Pacific Salmon Commission Seminar Series on Environmental Change in Pacific Salmon, the view from both sides of the border. Topic for today's seminar is predation on salmon. And it features presentations by Dr. Brad Hanson from NOAA Fisheries at the Northwest Fisheries Science Center and Dr. Strahan Tucker from DFO at the Pacific Biological Station in Nanaimo. My name's John Holmes, and I'm the Canadian co-chair of the technical committee, and I'm pleased to introduce this 12th seminar in our series. And so I think what I can say now is that this is a long-running series, and we hope it continues to run. So as I said, our seminar today is on predation on salmon, and in this case, it's by marine mammals. And predation is such a broad topic in current salmon assessment and management. There are a variety of lenses through which it can be viewed. For this particular seminar, we've chosen to focus on marine mammal predation on salmon, which seems pretty clear and maybe not all that complicated. But when you look at our seminars for today, there's a bit of a dichotomy here. They're at opposite ends of the predation spectrum. So Brad Hanson's seminar on endangered predators and endangered prey, southern seasonal diet of southern resident killer whale, is looking at the benefits of predation for an obligatory predator on salmon, but it's an important species of concern or conservation concern. And so we have to consider that in whatever salmon management we're implementing. If you look at Strahan Tucker's seminar on do pinnipeds target juvenile salmon of conservation concern, a case for greater spatial and temporal balance in diet sample collection, it's coming from a perspective of whether or not pinniped predation is actually a potential problem for salmon conservation. And I know there are strong opinions already on whether it is or it is not. But I hope when you watch the presentations today and engage in the question and answer session, which I'm expecting will be quite lively, that you'll get some really useful insights into marine mammal predation on salmon, and it may even change your mind, or at least shape your opinions that are generally quite well formed on the topic. So before we go any further, I'll introduce you to our speakers. So Brad Hanson, and you should be able to see his picture on the screen, is an ecologist with NOAA Fisheries, and he's currently studying foraging and habitat use of southern resident killer whales and health assessment of harbor and doll porpoises. Brad joined the Northwest Fisheries Science Center in April of 2003. He previously worked as a wildlife biologist at the National Marine Mammal Laboratory in Seattle. Brad received his PhD from the University of Washington, where he worked on the development of improved tag attachment systems for small cetaceans. He also holds an MSc in fisheries from the University of Washington and a BA in zoology, also from the University of Washington. Strahan Tucker is the head of the DFO Pacific Region Pinniped Research Program at the Pacific Biological Station in Nanaimo, and has been the head since 2018. The program's been rapidly expanding, and it's gone from 1 to 4 core staff, and it provides population assessments and fundamental research on Pacific pinniped species in Canada. The main research focuses on marine food web ecology of upper trophic level animals, including trophic interactions, distribution, bioenergetics, and alternative foraging strategies related to ontogeny and life history of meso and top predators, as well as links to shifting climate, infectious agents, and condition. He's working with a variety of programs within DFO to accomplish these goals, including the High Seas Salmon Program, Program for Aquaculture Regulatory Research, the Molecular Genetics Lab, and the Basin and Coastal Scale Interactions Group. Sean completed his PhD in 2007 at Dalhousie University in Halifax, so East Coast, where he focused on the implications of diet selection for energy gain and storage in harp-hooded and gray seals through the application of biochemical techniques, primarily fatty acids and stable isotopes. He also holds an MSc degree from McGill University in Quebec, which focused on juvenile salmon feeding energetics. So the plan for today is to hear about pinniped diets from Strahan, followed by predation of salmon by killer whales from Brad. After the presentations have concluded, there will be a short break, probably about 5 minutes, and then it'll be followed by a moderated question and answer session. And Laurie Wykamp and Marissa Litz will be your hosts for that moderated Q&A. I anticipate, as I said earlier, that this will be a lively Q&A because there are strong opinions on marine mammal predation on salmon and what to do about it. Please use the Q&A feature in Zoom to pose your questions to speakers at any time. As you're watching the presentation, you can actually upvote those questions posed in the Q&A feature, and the moderators will choose the highest voted questions to pose to our speakers. If there are more questions than we can answer in the time available, we will endeavor to get written responses from our speakers, and we'll include those responses in the seminar package that's produced at the end. Thanks, and with that, we'll get on with the presentations, and over to you, Strahan.
Great. Well, thank you for the invite. It's a pleasure to be here today. So I just wanted to start by acknowledging my immediate colleagues here whose expertise and effort have contributed to this data set, which I'll be presenting today. And obviously I'm here today, as John outlined, because pinnipeds are seen as drivers in the decline in salmon abundance and average early mean survival, particularly for Chinook, coho, and steelhead salmon. So just by means of introductions, just a brief overview, there are 5 species found here, but only 3 are really obvious and seen in coastal waters. And these are, of course, harbor seals, stellar sea lions, and California sea lions. The other 2 species, northern elephant seal and northern fur seal, are found offshore, often well off the shelf break. So to varying degrees, these species utilize both pelagic and benthic habitats, from rivers to river estuaries to the oceanic zone beyond the continental shelf, representing just a broad range of foraging strategies. And each of these species tends to be opportunistic in their diets, exploiting seasonally abundant prey and feeding on a mix of pelagic forage fish such as herring and benthic fish such as rockfish. However, explicit diet data for these species, particularly in British Columbia, are sparse and have been taken pretty much opportunistically and irregularly, so no systematic collections. But really, little is known about how key prey are partitioned both within and between species over varying spatial and temporal scales. So, of course, just to recap here, pinnipeds are thought to be driving dynamics of salmon population trends. This has implications for resident killer whales, as I'm guessing we'll likely hear subsequently, either because populations are reduced or either through direct competition for food. So today I'm going to focus on harbor seals in the Salish Sea, although we are accruing other data on other species to address issues elsewhere. So just to, as background, there was some updated diet work employing a novel DNA metabarcoding approach to estimate diets for harbor seals in the Strait of Georgia. And as stated explicitly in the introduction, Thomas et al. Specifically focused on estuaries of salmon-bearing rivers, entirely because previous research in the region indicated that salmon predation by seals is most intensive near river mouths. And I think the impetus here was kind of on method development. So the estuary and harbor seal hollout sites included Cowichan Bay, the Fraser River, and the Comox Bay. And for comparative purposes, scats were also collected from a rocky reef hollout site, Belchain specifically. Now, while a majority of seals in the strait do occupy such hollouts, any use of this particular site as a non-estuary index site is questionable, really, since it is directly across and washed by the Fraser River plume, of course, the largest salmon-bearing system in the region. And as we know from decades of salmon trawl survey data, the CPUE is very high in the plume, CPUE of salmon. So really, despite the limited spatial and temporal scope and diet estimates potentially skewed towards salmon, these results, so 2 years of data from 4 sites in the Strait of Georgia, were nonetheless incorporated into estimates of harbor seal impact, primarily on Chinook salmon in other areas, namely Puget Sound and the entire west coast of North America going back 40 years. So, I think there's some further issues with this, I would say, blanket application of these diet results. So, basically the Strait of Georgia has the highest densities of seals in BC and I dare say the coast. But we estimate that less than 10% of seals reside within proximity to estuaries, about 10 kilometers from a river system. Now we know that harbor seals have a very restricted foraging range anyway, but here through our recent telemetry deployments, ranges actually seem far less than reported in the literature globally at around, less than 10 kilometers. So, what is borne out continually in the literature is that diets of pinnipeds tends to be variable in multiple scales in both space and time. And furthermore, within a species, diets also tend to vary amongst demographic groups with differences between size or age classes and sexes. And all of these sources of variation in diets have also been identified in harbor seals, even harbor seals from the Pacific Northwest. So there are potential biases in the estimated total amount of salmon consumed. So in 2015, DFO initiated new research to provide a more systematic update of the temporal, whether that's monthly or annual, and spatial, contrasting river, estuary, and non-estuary sites, basically trying to look at the variation in pinnipeds' diets. And, we are using a dual approach for scat analysis, exactly the same methodology as in the initial work. So hard part identification done by Pacific ID provides information about the size of fish consumed, while DNA metabarcoding done by the DFO genetics lab under Christy Miller provides us discrete estimates in a very, very powerful method of the species presence and their proportions in the diet. Of course, we acknowledge there are biases with the method, and in fact are working to calibrate and validate the approach further, but relative contrasts are appropriate. So just to recap here, I've just plotted out the sampling sites for, I'm just calling it the pre-2015 work done between 2012 and 2014, where we had almost 1,500 scats collected at different sites. I've highlighted the bell chain here in the red diamond. Currently in their metadata, this site within the COMAX is also identified as a non-estuary site, but I think that is questionable. So again, in 2015, DFO initiated a broad scale sampling program at multiple haulout sites, but really collecting scats is kind of more of an art than a science. Some sites just yield more scat just based on conditions or the the geography, which, for lack of a better word, of the rocks and so on and so forth. So most of our samples come from sort of a bunch of core sites within here, and we collected over almost 3,000 samples have been analyzed, and samples were collected on a monthly basis over multiple years from spring to fall. So here I am just showing the mean occurrence of different salmon species between estuary, non-estuary, and that bell chain site in the initial collections. And you can see that salmon occurs in between 10 to 40% of scats, with not much difference between the site types. And obviously I don't have a lot of time to explore these in greater detail, but there is seasonal and annual variation that I'm not showing you. And here are the mean occurrences in the later data set. So overall, these tend to be lower than the previous sampling, ranging between 2 and 20%. And depending on the species, differences between site types. So bell chain is typically higher, and for chum and chinook, occurrences are higher in estuary versus non-estuary sites. So here I'm showing, in a somewhat similar manner, the diet proportions of different salmon species, contrasting the 2 collection periods between site types shown in the different colors. So with the exception of chum, coho, and sockeye at bell chain, as a mentioned, there are discrete seasonal and interannual differences that I'm not showing. The mean diet proportions are higher in the initial sampling period and higher in estuary versus non-estuary sites. And I'm focusing on the means here because it is these which have been propagated through to estimate pinniped impacts. Now, briefly touching on other sources of diet variation. For a subsample of individual scats, we had the sex of the animal determined by genetic means. So again, males and females in different colors. And here I've teased out some of the seasonal effects. So you generally see increasing salmon consumption between spring and fall. But on average, males consume about 9 times more salmon than females. But that being said, there is a bias towards male scats in our collection. So for every female scat, we have about 3 male scats. These results have similarly been borne out in the pre-2015 data set. So we think this sex ratio skew is related to hollow behavior, is not a true reflection of what's happening in the population. It's just, I think males tend to hollow out higher, and there's just more probability that scats will be retained and we come along and collect them. But that being said, we have no independent check of the population sex ratio. So switching gears to the hard part analysis, again, this provides us information on the size of salmon consumed. So here, all bones are considered, it's not just otoliths, for example, and they cannot distinguish species, and there are overlapping size categories. So this plot shows the classification of 300 or so salmon bones into size categories between estuaries and non-estuary sites across seasons. So out of about 2,700 scats examined, salmon bones were present in 15% of those, 11% from estuaries. Obviously, we see a higher occurrence of salmon in estuaries, but really only, I think it was 22% of all estuary scats had salmon, and only 8% of all non-estuary scats had salmon. So about a quarter of the salmon bones are within a very generous designation here, smolt size class, up to 30 centimeters. And, you know, most of the bones are retrieved in the fall. But what is interesting is that there's a large component of larger than presumed salmon in other times of the year than fall. So that is, they're consuming other age classes, not simply smolts in the spring or adults in the fall, as has been assumed, often assumed in the biogenic models of predation impact. And this is important because the mean salmon size is the denominator in estimating the total number of salmon consumed. And like I say, we know from decades of trawl survey that there are overlapping age classes of different species present at all times of the year. So this really shouldn't be a surprising result. So here are the straight up means for the different salmon species in the diet. I'm just contrasting the 2 collection periods. So which are between 2 and 7 times lower in the later period than those previously reported. Now, I think a part of this is a potential sampling bias, as I've inferred, but as we know from targeting in on specific sites like the Cowichan River, for example, salmon was just higher in those initial years. So our sampling is far from perfect, but I think what we can conclude is that it matters where and over what time period you sample, and especially if you're going to go take this data and make large inferences with it. This is not to say seals don't have an impact on salmon, but we need to generate empirical data to identify where and when that occurs to understand the implications for salmon population trajectories. So by expanding our spatial and temporal timescale, we found that the mean percent of salmon is less than previously documented, that the size of salmon consumed is greater than previously assumed. And if you just follow that through under the published bioenergetic approaches of estimating the total amount of salmon consumed by seals, these would result in a reduced impact. And this is, analogous to recent work published by Nelson et al., where first actual diets were estimated in Puget Sound from scats, and second, when the prey size was estimated from the otoliths, you actually recovered from seal scats. Seals were consuming larger fish than presumed, and this subsequent estimated impact decreased by 71% for Chinook salmon. So here's just a bar plot of the mean diet proportions by fish group, showing the seasonal means and diets between locales, and you can just see how variable it is. So salmon is a low frequency, low proportion prey. The diets are both diverse. We've identified over 60 different prey species, but are also dominated by hake and herring between 40 and 75% of the diet. And this brings me to another interesting dynamic of species co-occurrences. So here I'm looking at salmon and hake, a presumed predator of salmon. So just looking at the DNA diet proportions in a diet, and here just assuming that a scat equals a meal. So we had 470 occurrences where salmon is present in scats. Of those occurrences, hake is also present at 43%. And 75% of the time, it's at proportions equal to or greater than salmon. If we switch to hard parts, we had exactly 323 occurrences in scats of small salmons, less than 15 centimeters, in which 28% of those co-occurred with a large hake. So we had a total of 107 large hake co-occurrences with salmon. Therefore, we had 85% of those are with small sized salmon. So I just think this raises some questions about the possibility of secondary consumption. That is, maybe some of the salmon we see in seal scats were initially in hake scats. So we are really trying to address this issue, which has really remained unaddressed over the application of scats, with feeding trials with captive sea lions, and also various experiments to refine the DNA methods so that maybe we could identify benchmarks for including a prey item in a scat or not. So harbor seals are generalist predators with broad and diverse diet. So we are undertaking work to understand their functionality within ecosystems and the ecological interplay amongst species. So rather than simply focusing on the means, we are attempting to explore the variation. Just a word about scats and the methods we employ. Obviously, they're easy to obtain. They generate species composition and size data. We can extract some information about the animal itself, for example, the sex, but really they're only a short-term snapshot of the diet, and they may not capture diets for far-ranging animals. So for example, you can have sea lions feeding out at sea for extended periods of times, and that diet would simply not be captured through scat analyses alone. So you might be missing a large portion of its dietary space. There are other biochemical methods of diet estimation, which are more integrative over weeks to months. And these are fatty acid analysis, where you examine the fatty acid composition of lipid stores. Basically, you are what you eat relative to your prey, and stabilized tooth analysis. So I can talk about this further, but I'll in the interest of time, I'll move on. So last year, we developed a biopsy system for sea lions using crossbows. It's easier for us to obtain biopsies of skin and blubber for these analyses from harbor seals by simply capturing them directly. And we've been undertaking multi-species sampling along key salmon migration routes and killer whale feeding grounds. So our goal is to estimate competition between pinnipeds and killer whales for salmon prey, but also to understand how diets are partitioned among species, groups, across habitats, areas, and over time. I've shown you some of our harbor seal data from the Strait of Georgia. We also have sea lion collections there. We expanded this core sampling in 2018 to include other key migration areas for salmon. But each of these areas also represents a comparatively unique oceanographic and biological system. So it'll be interesting to compare the dietary niches in the community of pinnipeds there. We were sort of cut short in 2020 with COVID. We resumed all this collection last year, but also bringing to bear these other integrative complementary means of dietary estimation, where we hope to gain a fuller and more integrated picture of seal and sea lion diets. So I will just finish off with an acknowledgement slide, and thank you very much.
Thanks for that, Strahan. And now we'll hear from Brad Hanson on southern resident killer whale diets.
Well, thanks very much for the introduction and whatnot, and happy to be able to talk about endangered predators and endangered prey, the seasonal diet of southern resident killer whales. I wanted to first acknowledge all of my project collaborators. This is an effort we started about 20 years ago, and we're still at it, looking at different avenues. But it couldn't have been done without all of these people, and well as a lot of volunteers. So a little bit of background first. Southern resident killer whales are a small fish-eating group of killer whales that occurs primarily in the Pacific Northwest. It's comprised of 3 pods, J, K, and L. In the map on the left, the purplish area from essentially up off that yellow area in Canada is the northernmost extent of their occurrence, and that extends clear down to Southern California, or Central California, I should say. There was a 20% decline in the southern resident killer whale population that occurred between 1995 and 2001. And this led to a petition to list this population under the U.S. Endangered Species Act in 2001. And you can see that decline in the graph there in the middle. And consequently, in 2005, southern residents were listed as endangered under the ESA. And I should mention that Canada actually had listed them under their Species at Risk Act in, I think it was 2001. So they are protected in both countries. So when the U.S. lists the population of the ESA, there's a couple of things that have to happen. One is the development of a recovery plan, and that's designed to understand and address the risk factors and data gaps limiting the recovery of the population. And one of the risk factors was prey availability, but one of the challenges with trying to determine prey availability is you got to know what they eat first. So that was one of the things that we were, set out to try to establish. And then the second thing was trying to, well, I should mention that winter distribution was also ranked as a primary data gap. And so not only do we have to figure out where they were in the winter, then we had to figure out what they were eating there. And then we're also responsible, the second thing that we need to do is designate critical habitat for the population. And this is also a species in the spotlight in NOAA fisheries. And there is a 5-year action plan that gets updated every 5 years. And number one thing on the list was target recovery of critical prey. So some information about some of the previous studies that, that on killer whale diet. These whales were first photo ID'd back in the early mid-70s using a technique that Mike Bigg developed. And then subsequently Ken Balcom took that particular technique and established that the composition of the southern resident community back in about the mid-1970s. And there were anecdotal observations of them eating salmon, but it was, the picture at the right is not something that we typically see. That particular photo was taken by Astrid Ginniken at the Center for Whale Research, and her comment was to me when she took that photo, she said she'd been waiting 20 years to get a photo like that. The second thing was that to look at whale presence versus salmon catch data. And this is essentially only inferential, and it was basically guilt by association. And Jim Heinrich-Barand had published a paper in 1988 that suggested that given the, sometimes massive runs of sockeye salmon returning to the Fraser River, that particular species would be suggested to be of particular importance to southern residents. And then finally, John Ford published a paper on stomach contents and surface prey event sampling in 1998. And that was really sort of our first look at what these animals were eating. And John had looked not only at northern residents, but also at southern residents. And in the case of southern residents, there were 5 stomachs that had been recovered, and 3 of those contained Chinook bones or flesh. And they also collected 28 samples from fish predation events, and 7 of those contained Chinook salmon, but also a wide variety of species of salmonids and some non-salmonids. And this is really sort of our first indication that Chinook, were potentially a preferred prey item. But one of the things that we were a little bit skeptical about was just that these samples have been collected over almost a 30-year period. And so how representative that was, was unclear. So beginning in 2004, several of us started a study looking at southern resident killer whale foraging. We were trying to do two things. One was to collect behavioral information associated with foraging by the animals. And so we basically followed in the fluke prints of the whales. And this was a technique that was very similar to what John Ford had doing, because we had been skeptical of what John's results were, and we felt that it was important to try to replicate the methodology that he used. And so part of our objectives were to define foraging behavior better, because one of the things we found that it could be quite subtle. Although the whales do chase fish around at the surface, it doesn't happen all the time. And in fact, in about 30% or so of the predation events that we were able to document by, following the fluke prints, it can sometimes be quite subtle. And there were just behavioral cues, that changes in speed and direction and associations with other whales that essentially tipped us off. But the second thing we were doing was to collect remains from predation events and primarily scales, and that's what John had collected. And so this is just a photo of one of the salmon scales that was collected in net. And the objective here was to try to obtain a contemporary sample of sufficient size to accurately determine prey preferences. Oops. But one of the things we had noticed was that from a behavioral standpoint, is that a lot of times the whales would just converge, and this was likely due to prey sharing. And this is something that John and Graham Ellis had documented in their 2006 paper in northern residents. But one of the other things that we found besides picking up scales was sometimes we had bits of tissue, as shown in the photo on the left. The other thing that we were sort of surprised to find was that we were finding a lot of other stuff in the wake of whales. And one of the primary things was feces. We also collected mucus samples as well as regurgitations. And fortunately, at the Northwest Fisheries Science Center, we have a pretty robust genetics program. And so we were able to apply some of the techniques that they had developed in trying to look at essentially use these tissues as well as feces to determine diet. So the first 4 years of our study, and this is published in a paper that's cited down at the bottom here in 2010. We basically were collecting samples from the whales in inland waters of Washington, and then John Ford had contributed some of the samples that they had collected out in the Strait of Juan de Fuca. And so essentially what you're seeing here is this plot showing that the blue bars are Chinook salmon. And this was, exceeded about 80% for all the months with the exception of May, where we had a relatively small sample size. There were actually few sockeye, and no pink salmon showed up in this sample, and only one non-salmonid fish. So one of the questions that I had asked at the time was that we collected our first samples in 2004 was, where are these fish from? And I posed this question to Paul Moran at our lab, and this was in 2004, and he says, well, give us about a year, and he says, we'll be able to tell you, because we are working on a coast-wide standardized microsatellite DNA database for Chinook salmon. And so this was published in 2005, and essentially this is commonly referred to as GSI. And so what we were able to find out from this, looking at the seasons here in this plot, was that The Fraser River is of primary importance for diet southern residents during the summer. It's essentially kind of a mixed bag in the spring, although that was somewhat of a small sample size, but virtually all of the samples in June, July, August, and September for both the Straits and of Wanafuka and the San Juans were similar. And what we saw was the upper Fraser was really important in June. In July, the middle Fraser. By August, it was the South Thompson. And then by September, it was essentially the lower Fraser. So each of these different parts of the Fraser had a very important seasonal component in the diet of southern residents. So one of the things that we were interested in trying to do was to try to figure out how might the feces that we've been collecting be used to contribute information on diet. And this took us quite a number of years, but by 2016, Mike Ford at our center published a paper that essentially was a comparison of the fecal data we'd collected in the summer to the scale data that we'd collected. And so what we were able to get out of this was essentially a proportional contribution of by diet by species. And so during this summer period, which was basically about the same as what we were looking at for scales, we found that salmonids actually made up 98.6% of the sequences, with a little bit of halibut and herring mixed in for non-salmonids. Again, Chinook, the proportion of the diet was almost identical to what we saw with scales. It was about 79%. Coho were relatively common, 15% of the sequences overall. And there were 3 other species, chum, sockeye, and steelhead, that made small contributions. The early summer samples were predominated by Chinook in all the 5 years that we looked at. And then midsummer samples were also mostly Chinook, but in 2 of the 5 years, sockeye accounted for about 12 to 18%. And then the late summer samples had the lowest proportions of Chinook and varying fractions of coho. And the timing of this probably was due, or the variability of this was probably due to the timing and collection of samples. In looking at this a little more closely, it seems that particularly samples collected in the latter part of September are typically predominated by coho. And so depending on when we had scheduled our field season, this probably affected some of the results. So one of the things I did want to mention is that summer distribution of southern residents in the Salish Sea is changing. This is a plot from a recent publication of my colleague Sheila Thornton and her co-authors, showing the distribution of occurrence of southern resident killer whales and during the summer here in the last few years. And basically, the area off of the southwest side of San Juan and up off the Fraser River had been used quite a bit. That's really changed. The area out at Swiftsure Bank now is used predominantly during the summer by some of the southern residents. And I know that Sheila is working on diet composition from some of the samples they've collected there. And so we're excitedly waiting to see what sort of results they're getting, to see if it's different than the samples that John had collected and contributed to our 2010 paper. What about fall diet? So October to December, and essentially looking at what the whales were eating while they're in Puget Sound. And again, we used feces and fish scales and the same focal follow technique. The map on the left shows where we collected samples. The whales basically use the main basin of Puget Sound. They also go up into Possession Sound and Saratoga Passage. That's an area that I'm still interested in trying to collect samples from to see what they're eating up in that area, but we just haven't had that opportunity. But when you look at the scales and tissues in the top right panel, you'll see that it's dominated by chum. Chinook actually makes up about 20%, and then we also have steelhead and coho. But when we look at the quantitative fecal DNA in the bottom panel, you actually see more Chinook than we do chum salmon, and then with a number of other things sort of mixed in. The difference in the feces, well, the main thing I wanted to point out was that there's just a lot more diversity showing up in the diet with feces. And the fecal samples likely represent species that are consumed outside of Puget Sound. The whales, a lot of times, will come in and spend a couple of days, about every 10 days to 2 weeks between latter part of September and the early part of January. So it's a situation where there is a passage time through the digestive tract that it actually isn't all that well known. It could be anywhere from a few hours to maybe a day or so based on some of the work that's been done in captive display facilities. So when we look at the GSI from the scales and tissues, not surprisingly, fish consumed in Puget Sound are predominantly fish from Puget Sound, but there's actually about 20% of that were from Canadian origin. And there was even a smattering of samples that fish from as far south as the Central Valley and as far north as the Upper Skeena. So what about winter? And as I mentioned before, this was a primary data gap in the recovery plan. We had a workshop in 2004 that identified techniques to try to address this data gap. 4 of them are listed here. I'm going to talk a little bit about the sighting network, satellite tagging, and then ocean class vessel cruises. So as I mentioned before, the whales have been photo identified since the mid-1970s. The first documented documentation of them on the outer coast was actually in 1982. And between 1982 and 2005, when they were listed, there were actually only 33 winter and spring sightings of the whales. Between 2006 and 2012, we supported a coastal sighting network, and that added about 42 additional sightings. So although that it did provide some information, we did need a lot more information about where the whales are spending their time. So we employed a satellite tagging project where we use these limpet tags shown here on the left, and these were projected onto the dorsal fin of the whales, and we were able to follow them for periods of up to 90 days. And so this is a couple of plots of the duration of occurrence for a few of the whales. In the left panel are K and L pods. Ks and Ls tend to use the outer coast area from up at the north end or the west entrance of the Strait of Juan de Fuca, and they range all the way south to Monterey Bay. The satellite tags actually, the two years that we had them tagged, 2013 and 15, they actually only ranged down to Point Reyes. They didn't spend a lot of time down there. Most of the time, as you can see, was spent off of the Washington coast, particularly off of the area between Westport and the Columbia. Now, J-Pod, on the other hand, does something a little bit different, and that's in the right panel. They basically sort of split their time between the west entrance, the Strait of Juan de Fuca, and the northern part of the Strait of Georgia. So one of the things we did besides the satellite tagging is we combined it with our ocean class vessel cruises. And so we took the small boat along, used satellite tags locations to locate the whales, launched the small boat, and then proceeded to collect samples. And when we take a look at the feces sort of over time, what we tend to see is basically a lot more diversity in the diet in the winter in terms of, and that's in the left panels, both on a diversity in per fecal sample as well as the number of species that are seen. And this is both on the outer coast and in the Saley Sea. And we're just, like I say, it's seeing a lot more diet diversity in fall and winter. You know, in the right panel, essentially you're looking again at the whole year. Chinook decreases in early fall, coho increases then. In the late fall, chum becomes much more prevalent. And then winter is a much more diverse diet, including lingcod, steelhead, skate, and flatfish. So this is just a summary of the seasonal diet that we've been able to establish to date. It is dominated by Chinook. The predation event samples and the feces do show similar diet patterns, but feces does show more diversity. Chinook are the primary prey species in the summer diet and originate mainly from the Fraser River. In the early fall, coho increase in prevalence in their diet. Chum predominate over Chinook in the fall diet in Puget Sound. There's not a lot known about fall diet outside of Puget Sound yet, although the preliminary data do show at least some sablefish being taken. We actually don't know a lot about the winter-spring diet of J-pod in their Saley Sea range. And then finally, although the coastal winter-spring diet for K&Ls is dominated by Chinook, particularly from the Columbia River, Central Valley, and Puget Sound, the diet is much more diverse than in other seasons. And then finally, some of the future modeling efforts are going to attempt to inform the degree to which certain species and stocks of southern resident annual diet are impacted by their predation. And with that, I'll put it up for questions.
Now to the part of the seminar I always enjoy, which is question and answers. So Risa and I are going to alternate questions to our two speakers, and again, those were fantastic talks. So we'll start off with a question for Strahan from Gary Morishima. He said a couple papers from UBC related Fraser sockeye declines to stellar sea lions, and wondering if you had any comments on that.
Yeah, thank you, Gary. I believe you're referring to, it's a perspective paper where I believe, yeah, so they attribute stellar sea lion predation during the return migration as resulting in the decline of specifically Fraser River sockeye. I believe that's the paper you're referring to. But I believe this, it's clearly stated in the paper that they assume upwards, they have to make an assumption of upwards of, I think it's like 40% of sockeye in the diet for stellars over that whatever month-long return migration period, and that they really stress that it's not actually based on any empirical data, but it's simply an assumption. So, you know, I didn't show our sea lion data, but, you know, again, most salmon, with the exception of chum, and particularly chum in the fall, within sea lions is a low frequency, low proportion diet item. Chum can make up a substantial portion of the diet, that being said. But at this point, I've never seen empirical data that would support that assumption of, you know, these super high levels of sockeye in the diet. That being said, you know, we do have samples from that area, but we could certainly expand our focus and our work. And indeed, we've put in funding proposals to do such, but it certainly outlines a very testable hypothesis to pursue that. So hopefully I'll answer your question there.
Thanks, Strahan. So the next question is for Brad. And it comes from Gary, and he is asking, so one measure under consideration, well, actually it's more than under consideration, it's being applied, but to help southern resident killer whales is increasing hatchery production. So can you comment regarding how this might affect prospects for the endangered killer whale?
Yeah, I mean, much of the information that we've collected has fed directly into the priority prey work that the regional office has been using to develop hatchery mitigation measures. And so we are hoping that that these increases in production will in fact result in whales, essentially helping with recovery. I mean, one of the things that I am interested in doing, and we're a little bit challenged right now with funding and whatnot, is just to try to get out more in the winter. We are starting to see, some increase in occurrence, at least by JPOD. Although it's anecdotal at this point, the question is that what are they feeding on when they're coming in at some of these other times of the year, other than just the fall months. And our hope is that, these fish that are being increased in some of these hatcheries are essentially being attractive for the whales to stick around more.
Great. Question for Strahan from Ed Johnson. He says, are you getting any codeware tags in your scat samples? And can you use that to help identify what stocks you've got? Or are they too small to really see?
Yeah, that's a really good question. And we have not screened our scats for codewire tag, so there's a possibility there. I know other groups have gone out. There's a couple of haulouts sort of in front of Couch and River, and they just sort of scanned the whole area for codewire tags, and I've identified some there. And and that's also the case too for heron nest and so on. But no, it's a good point, and perhaps something we could easily integrate.
I certainly know how many I've lost.
Yes.
Them, so they're very small and
yeah,
easy to work with. But yeah.
So along those lines about stock ID, this is a question that we could put out there to both of you. Maybe start with Brad. But has there been any steelhead stock ID in either of the pinniped or killer whale studies?
Well, I know that we would like to look at that. I'm not real sure what the status of GSI on steelhead is. I don't know if maybe Lori knows more about this than me or somebody else on the call.
There, Don Van Dornick has a steelhead baseline, and actually there's one right now being developed for our high seas steelhead work. So yeah, that is an issue, is that there's not a good coast-wide baseline right now. So
right, and I should also mention that Kim Parsons in our genetics group is also working on trying to use some of our fecal samples for stock ID, and Don's been working on that too. And it's a bit more challenging because the DNA is pretty beat up by the time it comes through the digestive tract. And I think that, we've been able to have like about a 10% success rate with it, but it's an area that, with genetics always seeming to have new techniques available, it's an area that we are trying to pursue, but just like some of our prior work, it takes time to develop these techniques. And so same thing with coho. We're seeing a lot of coho being taken, seasonally. That would be all, particularly given where they're taken up in the San Juans, would be of interest to know which stocks they're actually coming from.
Strahan, do you want to comment, especially with the high vis, the steelhead and harvester interactions in Hood Canal?
Yeah, so I'll just comment that, in our, I was looking this up because I saw the question earlier, in our data set for steelhead, we had 2 observations of steelhead out of those 2,700 scats, and they were both from the Couch and River. But yeah, and just on this question of stock ID, it'd be super interesting. But for reasons that Brad outlined, it's not super high on our priority, just because we're kind of waiting for someone else to put some money and effort into it before we go there ourselves right now, just given limited resources. But yeah.
So we have question, again, it's for either Brad or Strahan. Maybe we can get both of you. Well, I guess this is a Strahan question. From Gary, he says the Washington State Academy of Sciences recently issued its report on pinniped predation in salmon. He was just wondering if you had any comments on the findings or recommendations. Were you part of that study as well?
No, I was not part of that study. And I had a cursory look at sorry, the cat just gone at the document. Sorry. But as far, we also held our own, and there were some bilateral workshops in 2019 and 2020. But as far as I could tell, there was no updated data presented at this workshop. So I think they came out with some different recommendations, but I wasn't at all a party to those recommendations. So.
And Brad, how about you? Were you part of the Washington Academy of Sciences pinniped predation in salmon study?
No, I was not. In a previous life, I had worked on that sort of thing, and now I just run the other direction as fast as I can.
Fair enough. So I'll pivot to another question for you, Brad. So this comes from Urs, and the question is, so interesting presentation. Surprised to see the relatively high predation on steelhead during the winter months. Is there an explanation why they don't feed on pink salmon?
Yeah, no, that's a really interesting question. John Ford actually had documented them feeding on pink salmon, but he noted that it was only the juvenile whales that were predating on them. John did a really good job with Graham in the 2006 paper outlining why Chinook are preferred prey item. And as I think I mentioned, one of the things that these whales do, because they are so social, is prey share. And so, Chinook are the biggest, fattiest fish. And again, it wasn't surprising to see them eating chum, which is the second biggest, fattiest of the salmon. So I think that, the whales are essentially, if given how much the energy they have to invest in predating, it's probably best to go for the biggest bang for the buck. And one of the other things too is just that, the behavior of Chinook is probably different. Some of the telemetry studies that John Candy had worked on showed that Chinook are relatively deep, and that's what we're seeing in our TDR work. Whereas a lot of the other salmon are more up in the photic zone, and they tend to school, which is an anti-predator strategy. And so that, they may see the whales, coming at them, and it just, the amount of energy it may actually take to catch these fish may not be worth it. We saw sort of the same thing talked about this in our 2010 paper, in that although the recreational catch fishery in Washington State up in the San Juans, there's a lot of blackmouth, which are smaller because they're these delayed release fish that are around about 8 pounds. Taken in the southern area, around the southern area of Rosario Strait, we have never seen a southern resident come down Rosario and take a blackmouth in that area. Not that they don't do it while they're in Puget Sound, but it does beg the question of, are the whales, avoiding these smaller fish, or are they having a harder time detecting them because they are smaller fish? We did, a number of years ago, we did a controlled study pinging different species of salmon, and this was done by Whitlow Au from the University of Hawaii and John Horn at the UW with Chris Jones from APL. And basically what they did was they x-rayed the fish. They got a look at what the swim bladder looked like. They pinged it with a 69 kilohertz ping, looked at the backscatter of that, and basically this is how the whales tell the different species apart. The experiment we really wanted to do, but there wasn't money, was we wanted to basically take these fish out, because this was done, you know, about 10 feet of water at the APL facility, was to go out off a floating bridge in Lake Washington, run the fish down to, you know, about 120 meters or so, squeeze that swim bladder down, and then see what that backscatter looks like. Because, you know, they just may not be able to see the, you know, detect these smaller fish.
Fascinating. Question for Strom. I think you've stirred a lot of interest in this whole issue of hake and salmon and primary and secondary predation. And so we have two questions regarding that. One was from Urs, and he said, have you looked at prey availability between salmon and hake during your research, in that you're seeing, you know, such a large hake signal? Does that correspond to pretty high hake abundances? And then as a rate related was just, you know, how do you tell apart whether the salmon was free swimming or had already been eaten by something else?
Yeah, so, you know, within the Strait of Georgia, there used to be a commercial hake fishery, and decades ago that's been shut down. And unfortunately, there is no groundfish survey in the Strait of Georgia. So we we actually don't have a true sense of what that hake abundance is explicitly, except, you know, through data like this, or, you know, other pelagic surveys or so. We know that there is an abundance of hake. So unfortunately, we can't make those direct ties here. I think, you know, as far as again, you know, there's a lot of issues with looking at scats, but I was just trying to raise the point that, you know, you do have these co-occurrences that might intimate there isn't a secondary prey issue. And I think people acknowledge that there certainly could be, but it's a hard one to get a handle on. Sorry, a couple trains of thought. So, you know, we are sort of doing, you know, through the DNA, you know, basically we've taken fish caught and we dissected out their digestive tracts, then, you know, profiled their, you know, do a genetic profile on what a herring looks like, you know, both with and without its digestive tracts, and just, you know, see what that looks like. And if you can identify, you know, cutoff points in your DNA yields that would say, well, we don't really believe necessarily that, you know, a prey item with this many reeds should be part of the diet. But, you know, it really is a tricky issue to get a handle on, looking at scats, fortunately. So I don't know if that answers your question, but.
Thanks, sir. And I actually thought that was one of the most interesting elements of your entire presentation. So there was, I don't know if you're aware.
It's inferential, admittedly, like, I don't know, but it just raises questions, I think.
So I don't know if you're, there's work that was done. It was hake diet analysis. Several thousand hake stomachs were examined, you know, off the Columbia River, and very, very few, like in a handful, like less than 5 salmon observed in those stomachs. So to be observing this in the pinniped dice would be fascinating. But.
Yeah, and I will say, like, the DNA method's so sensitive that our samples are extracted in a forensic lab because we're worried about salmon contamination, you know, because there's a lot of salmon work done in the department. So, I mean, you know, you think a fish, 100-gram fish has, you know, eats probably 1 to 5% of its body weight per day. So it has several grams of material in its stomach. Wouldn't be surprising if our DNA would pick that up. You know, anyway.
Thank you. So back to Brad. So we have a question from Cheryl. So it's more of a clarification question. So summer is presumably June 21st through September 21st. And she's asking whether the seasons are based on solstice and equinox, or, you know, some other criteria.
It's basically whale occurrence in different geographic areas. And that is like I was saying, it's sort of changed over time. It was fairly common in May for JPOD to be around in the Intersalient Sea. I mean, they're still there most of the time, but they used to spend a lot of time off the San Juans. And then usually by the end of the month, Ks and Ls would come in, and then they would be around until sometime in September or so. So we were basically defining it based off of area use by the whales.
Strahan, we have a question about the comparing the older and the newer data, in that were the mean values of occurrence of salmonids for the diet standardized to compare to the previous study? Is it possible that the decreased presence of salmon in the diet in your work is due to a lower abundance of salmon in the Strait of Georgia after populations are being impacted by the blob? And I was also wondering whether the anchovy invasion also may have impacted that there's alternate prey, more of it around, not just herring and hake.
Okay, yeah, so no, they're not standardized, they're just straight up means, but they are done exactly the same. But standardizing, I mean, to be honest, I think as Laurie will acknowledge, we have no idea how many smolts are actually entering the Strait of Georgia, so that's very difficult thing to do. So, but to your other point, I would, of course, couch and Chinook is a indicator stock for Chinook. And over those years of the study, those initial years, Chinook was very high in the diet and declined, in subsequent years. But interestingly, those years of, now that we have the data, those years of high abundance in the diet were actually, 2012, I think 13, were actually really enormously high years in terms of juvenile marine survival. So yeah, maybe the proportion in the salmon diet is an indicator of abundance. So presumably it's probably, I don't know, it's probably coupled to abundance. I mean, they are opportunistic predators, that's for sure.
So Brad, back to you. So we have a question. I don't know if you have the information off the top of your head to answer, but it's what was the approximate location that you sampled the upper Skeena Chinook in the diets?
I have been asked about this before, and I'm pretty sure it's up off the northern Washington coast. That's an area we saw a lot of diversity, and this is somewhat similar to some of the work that Ole Shelton has done in looking at coated wire tag recoveries. And I'm also looking at Laurie shaking her head yes, because I remember you talking about how, because I was really surprised that we would have a Central Valley Chinook supposedly show up in Puget Sound. And I think I specifically emailed and asked you about it, and you said, well, despite the fact that Chinook tend to have really good homing instincts, they wander around quite a bit. And I see you shaking your head yes, so I'm hoping that I'm remembering that correctly, because I think that was clear back when I was working on the first paper, 15 years ago.
Yeah, there's a lot of stuff. There's a lot of different salmon up in Haida Gwaii in that area. It's really fascinating, kind of a smorgasbord. We have now run out of questions, so I just want to thank you both again. That was absolutely wonderful. Actually, we've got a really quick question coming in from Craig Smith. He says, are the population of seals between the two study groups similar? I'm not sure which groups of seals.
I'm assuming over the time period, maybe?
Probably, right? Yeah.
Yeah. So I'll answer the question based on that inference. So basically, the population of seals has been stable at about 40,000 animals in the Strait of Georgia since I think the mid-1990s. Our last assessment was done in 2019, and we're maybe finally seeing start in a decline, but it's basically been constant.
Great, thank you. Yeah, it's impressive how many seals there are out there. thank you again to both of you for your talks and taking the time to come and talk to us. The recording will be available usually in a couple days on the Salmon Commission's, both the website and their YouTube channel. And then come back May 31st. We're going to be talking about climate assessments and salmon. We've got Michelle McClure, who used to be with the Northwest Fisheries Science Center and is now with PMEL, and Sue Grant from DFO, who will talk about some really pretty big studies they are both undertaking. So thank you again to our speakers, and thanks to everyone who was able to enjoy this talk, and we will see you in a month.
Thanks very much for having me.
Thanks, Lori. Appreciate it.